everyone. Today I will talk to you about the roles of hemodynamic forces in blood vasculature. I will explain what these forces are and how they affect the endothelium coming into contact with the pulsating flow of blood. Before I start introducing some technical terms, it is important to mention a few things to put everything in perspective. Blood vessels are a vital part of living organisms that allow for the transport of nutrients and oxygen to cells, as well as removal of waste products from cells and the supply of these cells with oxygenated blood, which is efficiently controlled by the pumping of the heart. Through this process of pumping blood to allow for an efficient circulation, arterial blood vessels are exposed to various hemodynamic forces, and these forces have an effect on the endothelial cells that line these blood vessels. These forces include shear stress and cyclic strain. And as you can see from figure 1 and 2, shear stress is the force that is applied onto a material when the material is subjected to torsion or bending along its axis. And in the context of blood vessels, it is the force applied along the endothelial lining as blood passes through. The greater the viscosity of the blood passing through, the greater the shear stress. Strain, on the other hand, is the change in shape that occurs in the material when force is applied to it. And when it comes to blood vessels, this strain is called cyclic strain, and it is the internal pressure of cells on the vessel wall that is provided by blood being pumped and exerting pressure on the circumference of endothelial cells. Shear stress seems to have a protective effect on the endothelium, as it decreases the chances of intimal lesions forming, as well as protection from atherosclerosis. Another example of the effect of shear stress on endothelial cells is the change in their alignment that follows the direction of the circulating blood, as well as morphological changes like elongation of these cells. Similarly to shear stress, cyclic strain leads to changes in endothelial cells like a change in orientation, for example cells aligned perpendicularly to the direction of the force, and inducing cell division and growth of smooth muscle cells. Shear stress plays a major role in developing vasculature, how our blood vessels develop. As previous research has shown that the shear stress is what drives the reorganization of one of the initial stages of vasculature in an embryo into the vascular tree that supplies all our cells with blood as adults. Scientists have found that it is shear stress that stimulates expansion and growth of vasculature, as low flow causes vessels to decrease in size and often to die out. Further studies have also shown that shear stress plays a major role in the maturing of the aorta and its branches, as in the developing embryo, two arteries connect the heart into the aortic arc, with the left one receiving higher blood flow and hence developing, and the right one regressing due to lower blood flow. So, shear stress also induces the expression of vascular endothelial factor, or VEGF, which plays an important role in developmental angiogenesis as well as maintaining of mature blood vessels in quiescent adult cells. Shear stress has been found to increase VEGF receptor 2 expression as well, and this signal is important for the protection of these mature blood vessels, as studies using VEGF receptor inhibitors show that these cells undergo apoptosis. Therefore, shear stress induction of VEGF with VEGFR2 signaling, and this reduces apoptosis and stimulates survival of these cells. Finally, I would like to add the fact that what we can see in figure 4 is the anisotropic nature of the aorta. Anisotropy is the property of material to not show the same stress-strain relationship when the force is applied in different directions. And in this diagram, we can see this as circumferential stress force applied gives a lower stress-strain curve than when axial stress is applied, indicating that the blood vessels show greater elasticity with axial force. Thank you very much.